This is a production of Cornell University.
Good afternoon, everyone. Thank you so much, Katie, for the introduction. And I would like to thank the chairs for the invitation. Unfortunately, I couldn't do the seminar in person, but I'm excited to be sharing with you today our ongoing research on the secure ecosystem engineering and design to mitigate the impacts of non-native fungal pathogens on managed ecosystems. So as Katie already presented, I'm an early career staff scientist at Oak Ridge National Lab. For those of you who don't know, I put the map. The lab is located in East Tennessee, and I put as reference Atlanta, which is 2.5 hours drive from Oak Ridge. And here's a picture of our scenic campus where cutting edge research meets natural beauty. I want to just give two fun facts about ORNL. So the lab has played an important role in the discovery of tennessine, adding a unique element to the periodic table. Also, it's the proud home of Frontier, which is the world's fast supercomputer. So at ORNL, we focus on delivering scientific discoveries to ensure the United States security and prosperity by addressing different challenges ranging from energy, environmental, and security challenges. Another fun fact, many are surprised to learn that at ORNL, we actually conduct fungal research because when they think of national labs, ORNL or other national labs, they think they just do like nuclear science, nuclear engineering, or material science. But the fact that we actually have a bioscience division is surprising for a lot of people. Actually, my colleague, Dr. Tomas Tarash, who I actually met at UW-Madison, he was also in the plant pathology department with Katie. And we both co-run the fungal lab and the fungal program at ORNL. Tomas's research mainly focusing on fungal interactions and biotechnology, whereas my research as a fungal geneticist and biologist focus on developing advanced genetic engineering tools to unlock the potential of fungi for sustainable applications. And here are pictures from our busy lab this past summer. The research I will be discussing in my seminar today is part of the Secure Ecosystem Engineering and Design, short for SEED, Science Focus Area. So in this SFA, we drive innovative science and technology to be able to understand and manipulate how microorganisms, can either be bacteria or fungi, which is our focus, are able to establish, spread, and how they are impacting ecosystems that are critical or of high importance for the US Department of Energy. So here's the outline of my talk today. So first, I will start by giving a brief overview about the pathosystem that we focus on within our CSFA, trying to further understand it. I will highlight the critical knowledge of gaps, mainly when it comes to understanding the molecular mechanisms responsible for the pathogenicity of our pathogen, and how we are. In my second part, I will talk about how we are driving innovative genetic research to be able to elucidate the mechanisms driving the virulence, but as well as developing genetic strategies to better control the spread of fungal pathogens. So first, why poplar? So poplars are vital crops for the Department of Energy. And because first they are like fastest growing tree species, but also because they serve as a foundational feedstock for producing biofuel, bioenergy, and other bio-based chemicals. At ORNL, we focus primarily at poplar, just because it has a fully sequenced genome. Actually, it was one of the first 3 genomes to be published, and it was this work has been done by a scientist at ORNL, and it made it to the cover of Science on September 15, 2006. But we also valued working with poplar due to its genetic tractability. So it has a transformation system and strong hybridization potential, which made it a prime model to be able to advance research efforts. However, this crop is limited to be used in industrial application, mainly due to the fungal diseases that are caused primarily by the pathogen Spirulina missiva, or known for the longest time as Septoria missiva, which is an ascomycete fungal pathogen. So this pathogen was first described in 1884 on Populus deltoides in New York State, but it has later been spread unintentionally by human activities and it was recently introduced and established into the Pacific Northwest of North America, and now it's everywhere. So in this figure on the right, I show the propagation of this pathogen that spread across North America, and we saw that there was like distinct population. So we have the southeastern US population, we have the US Canadian subpopulation, and we also have a genetically distinct British Columbia population. So I'm bringing this population right now because they will come up later in my talk, which we're gonna talk about them more in depth. But just note that we actually have 122 isolates of each of these populations that were collected, and we have the genome sequence for them. And that has really been very helpful to advance our research efforts. So this pathogen is responsible for the Septoria leaf spots and stem canker diseases on susceptible Populus genotypes. So while the leaf spots are mainly just of reducing the aesthetic values of poplar, the cankers are the ones that have the greatest industrial impacts due to the fact that a single infection can kill a whole tree and snap it in half. And that would reduce the yields and the use of poplar for industrial applications. Here's I'm actually also showing additional pictures that were taken by Tomas in our local Blount County Populus field, where most of our field experiments take place. So on the left, I'm showing how a diseased poplar look like, and healthy. So I put it in quotes, healthy, because although it is a resistant genotype, it can still be infected by spirulina, and that we've seen that several times where the pathogen is actually endophytically living inside the plant, but it does not cause diseases because the genotype is actually resistant to the symptoms and the disease development. Here's also other pictures showing canker disease propagation and how the cankers look like when it's a late stage canker. Just want to note that those cankers, although we were able to isolate spirulina from it, we cannot necessarily say that they are just due to spirulina, because we were able to isolate other pathogens that cause cankers as well. But just wanted to show you how a canker disease propagation look like. I believe a lot of you are already familiar with fungal pathogen classification. So as we know, pathogens, their pathogenicity have a spectrum ranging from biotrophy to necrotrophy. So biotroph are closely associated with living host cells. They extract the nutrients and grow and reproduce without killing the host. But that doesn't mean that they are not able to cause damage, which the great example is rust fungi that cause significant damage on crops. We have also the necrotroph that would rely on that host tissue to extract nutrients, and they release enzymes and toxins that will actually break down the cell wall and will kill the host cells. We also have, and the greatest example is botrytis. We also have a third class, which is where our spirulina pathogen falls in, which is the hemibiotroph pathogens, where they begin their life cycle as biotroph, but then later they will transition into a necrotrophic phase, and they will, into a necrotrophic phase where they will actually kill the tissue and extract nutrients and reproduce. So here's a brief overview about the life cycle of Spirulina missiva. So the infections actually starts in spring, where the pathogen release ascospores, where they will enter the poplar leaves through the stomata, and they will develop lesions 3 to 4 weeks after bud burst. Then during summer, there will be secondary infections where the spores will spread to other parts of the plants. They will enter the stems, the branches, and other leaves, and creating secondary infections. Then during fall, the leaves that are infected with the pathogen will fall off the ground, and then the sexual stage will begin with the formation of the sexual fruiting bodies, pseudothecia, and then the cycle will start all over again in spring. In terms of disease management, to mitigate S. mischievas infection, there has been use of fungicides, but just as a preventive approach, because once the pathogen is actually established, there was no chemical treatments that were shown to be effective. But as I mentioned earlier in my presentation, poplar has actually a strong hybridization potential, which actually made a substantial amount of research focused on the host site, either by identifying and planting resistant genotype or through breeding program efforts aimed at developing new genotype that can be resistant to the pathogen. Other management approaches aimed at preventing the spread of this pathogen has focused on biological control. So as I'm showing here in this first study in 2003, they were able to show that when they spray Streptomyces strains in the field, they were able to suppress leaf spot diseases. A more recent study in 2021, researchers were able to identify other Bacillus vilicensis strains that were also shown to have antagonistic activity against spirulina. But the problem of those biocontrol strategies is the limited longevity of the bacterial strains when they're used in plantation. The pathogen in this pathosystem has been overlooked for the longest time in terms of disease management. And this is mainly due to the insufficient understanding of the molecular mechanisms that drive S. mischievas pathogenicity. But also this intertwines with the absence of genetic manipulation tools. Where there's a lot of genetic tools, as I mentioned on poplar, for the longest time, we did not have any genetic tools to be able to manipulate this pathogen. So that made it our primarily objective within the CSFA is to develop CRISPR genetic tools for this pathogen that aims not only at advancing the basic research elucidating gene functions implicated in pathogenicity, but also at advancing applied research towards the development of CRISPR-enabled technology to be able to control the disease and ensure sustainable population management. So for the second part of my talk, I will discuss the progress that we've made so far into developing synthetic biology and genetic strategies for combating S. mischieva and mitigate its impact on poplar's plantations. And I will divide this part into three subparts. So first, I will talk about how we were able to develop CRISPR-Cas tool to genetically manipulate this pathogen. And then later, how we leverage these tools to uncover genetic determinants of pathogenicity and other genes that has ecological functions, and how we are taking this another step further to develop CRISPR-enabled gene drives for population control. But first, for those of you who are not familiar with CRISPR, CRISPR stands for Clustered Regulatory Interspaced Short Palindromic Repeats. It's a natural defense mechanism that was found in bacteria and that helped them in protect against viruses. But later, this natural occurring mechanism has been adapted for synthetic genome editing in a groundbreaking way and in a lot of organisms. So CRISPR is often referred to as the gene scissor. Why? Because it caused a double-strand DNA break in a specific sequence in the genome. So this tool actually is composed of the Cas9 protein, which will unwind and cleave the DNA double-strand break. And the second component of the CRISPR is the guide RNA. So it's the RNA component that is composed of the tracer RNA and the CRISPR RNA. So the tracer RNA, it will bind to the Cas9 protein, whereas the CRISPR RNA will hybridize to a specific region in the DNA. So to any gene of interest that we want to knock it out or delete it. So the CRISPR RNA is the part that we actually design based on our gene of interest. So the tracer RNA and the CRISPR RNA will fuse together to form what we call the guide RNA. And the guide RNA will form a complex with the Cas9. Then this guide RNA and Cas9 complex will go and will search for the sequence of interest that we want to knock out. And the nuclease domains of the Cas9 would be activated after the guide RNA hybridized to this specific region, and it will generate a DNA cut or a DNA double-strand break. Then this double-strand break that was created by the guide RNA Cas9 complex will be repaired in an error-prone way, generating multiple mutations, or what we call indels, referring to insertions or deletions, because they are randomly repaired. And most likely, those mutations will result in the gene being knocked out because its open reading frame has been cut in the middle and repaired in an error-prone way, containing a lot of mutations. So just to give you a brief overview about the advancement of CRISPR-Cas systems in fungal research. So this tool has revolutionized actually fungal genetic engineering in both yeast and filamentous species, enabling precise and efficient genetic modifications. And it has been used for various applications that range from genome engineering, primarily in yeast species, to create yeast libraries or to construct synthetic yeast chromosomes, as well as for metabolic engineering, transcriptional regulation. And we also recently saw that CRISPR has opened up new avenues for actually detecting and controlling fungal infections. In plant pathology specifically, the applications of CRISPR-Cas tools are also rapidly expanding. So these tools help improving plant disease resistance by targeting both the crops and the fungal pathogens, have helped deepening our understanding of plant pathogen interactions, and has also enabled the development of innovative diagnostic tools for early pathogen detections. So as a first step in our efforts, I worked on developing a CRISPR-Cas9 transformation system in our non-model fungal pathogen Asmisiva. So here I'm showing how the Asmisiva spores look like, and then how after digesting them with a mix of different digestion enzymes, how we are able to generate those bubble-looking structures that are called protoplasts. So those are cells without the cell wall. And then we use those protoplasts for the delivery of the Cas9 guide RNA complex to be able to generate the double-strand DNA break that I already talked about it. So as a proof of principle for the success of this tool in Asmisiva, I targeted an auxotrophic gene called PIRG because it allowed an easy screening on FOA plates in case when this gene is actually knocked out. And then randomly, I selected the 20 transformants from this transformation, and I did a sequencing of the region where the guide RNA is supposed to cut, and I aligned those sequences from the different transformants to the wild type, and I was able to see multiple insertions and deletions that actually resulted in the mutation of this gene, validating the success of this transformation and the success of this tool in Asmisiva. So now that we have developed the CRISPR-Cas9 tool in Asmisiva, I wanted to leverage it to identify genetic determinants of pathogenicity. And in fungal plant pathogens, pathogenicity determinants can be either carbohydrate-active enzymes that are involved in the degradation of plant cell wall, which are a key part of the pathogenicity of many fungal pathogens. We also have effector genes, which are secreted molecules that directly manipulate the host cell function and allowing the fungus to successfully infect the host and suppressing its immune responses. And lastly, we have secondary metabolites that are specialized molecules. So secondary metabolites, unlike primary metabolites that are not essential for the growth and development, but they provide advantage for survival of the pathogen, and they can act as virulence factors. And the production of those metabolites are usually regulated, as shown on the bottom right, by a cluster of genes. So these genes are clustered together, and they have different function. Some of them can encode for accessory enzymes and coordinate the synthesis and modification. Others encode for transporters that are responsible for the transport and the actual secretion of those metabolites. And we have also transcription factors that regulate the expression of those genes in the cluster. In our study, we focus on those two classes, identifying effectors and secondary metabolites that can be regulating the pathogenicity of Spirulina mucina. So to identify effector genes, we collaborated with Dr. Jared Leboldis at Oregon State University to conduct a transcriptomic analysis on S. mishiva during stem canker formation. So we collected RNA at 2 and 3 weeks post-inoculations with the pathogen. And this analysis have enabled us to reveal 70 genes that were differentially expressed at 2 weeks and 110 genes that were differentially expressed at 3 weeks between the stem cankers, like the RNA extracted from stem cankers, and the control, which is fungus growing on a media. And among those genes that were differentially expressed, the gene that had the highest expression at 2 weeks and the second highest at 3 weeks encode for extracellular protein 2, or short for ECP2, which is predicted to be an effector protein. It was characterized in other fungal pathogens and shown to be responsible for pathogenicity. It was found, as I mentioned, to be highly expressed during infection. And when we did an infiltration of this protein on poplar leaves, we showed that it invokes hypersensitive response in some of the susceptible genotypes. So using our CRISPR tool that we were able to develop, but this time, instead of having a random repair, we inserted, along with the CRISPR-Cas9 complex, a donor DNA that has a 1 kb homology arm that corresponds to the cut site of the guide RNA in the ECP2 sequence and contain a selectable marker, HPH, that encodes for hygromycin, which will allow us later on to select those mutants on hygromycin enriched media. So this is the structure of the guide RNA that was designed to cut in the ECP2 sequence. And after doing the transformation, we collected randomly 20 mutants. We did the PCR confirmation, and we selected of those positive PCR mutants 3 independent mutants that we performed whole genome sequencing to make sure that we don't have any Cas9 off targets, which is something that could happen, where the Cas9 would generate cuts in other parts of the genome and would also have mutations, not only necessarily at the targeted loci, but also in other parts of the genome. So we confirmed that in the 3 mutants that are independently selected, we did not have any Cas9 off targets. And we have a cut in the ECP2 sequence and a repair integrating the hygromycin selectable marker in those mutants. Later, we used those mutants and we performed pathogenicity experiments by spraying different poplar genotypes that represent a broad spectrum of susceptibility. So some of them are known to be susceptible to S. mishiva, where others were resistant. And as we can see here, by looking at the cankerous developments per centimeters, we were able to see that the knockout of ECP2 resulted in a significant decrease of cankers per centimeters in certain genotypes. But interestingly, those genotypes were the same genotypes that when we did infiltrations were also showing hyper response. So also when we looked at the leaf disease severity, we were able to see a significant reduction of leaf disease severity when the plants were inoculated with the Delta ECP2 compared to the wild type strains. So using this CRISPR-Cas9 tool, we were able to validate the function of the necrotrophic effector ECP2 that is involved in necrotic cankers formation on poplar systems. And this effector can actually serve as our first target for the development of the gene drive. But besides effectors, secondary metabolites were also our gene targets of interest, and they also play a key role in pathogenicity. So to be able to identify secondary metabolites that are potentially involved in S. mishiva pathogenicity, we used anti-SMATch, which is an online tool that is able to predict biosynthetic gene clusters in fungi. So when we ran this tool on the genomes of all 122 isolate of S. mishiva that we have from our collection, we were able to identify and that I'm highlighting in red here, a few biosynthetic gene clusters that were either truncated or absent in the British Columbia subpopulation that I talked about earlier, that is genetically different than the others population. And interestingly, when those clusters, clan 3030 and clan 3571, were absent in the subpopulation of British Columbia, we also saw that these subpopulation have also a reduction of cankers formation, which made us think that those clusters might be the good targets and might be implicated in pathogenicity. So I had two of my graduate students, Joshua Sparks and Cole Sawyer, who actually graduated last week, working on characterizing those two clusters and doing the knockouts of their backbone genes to validate their implications and pathogenicity. So the work on the clan 3571 is still in its earlier stages, and it's known to encode for an unknown metabolite. So it's a little bit more challenging than the work on clan 3030, which I'm going to be focusing on today, because this clan is predicted to encode for a metabolite that is known and identified in other fungi, but not in S. miseva, to be apenine-like biosynthetic gene cluster. So Cole has worked on characterizing this cluster that is predicted to be formed with 11 putative biosynthetic genes. And of those genes, as I talked earlier, there's a specific transcription factor, which is a gene highlighted in pink, APNH. So what is the role of this specific transcription factor? When a cluster of genes contain a specific transcription factor, it works as a switch. So when this gene is expressed, the rest of the genes would be expressed. And when it's knocked out or not expressed, the rest of the genes would be turned off, and therefore we won't have a production of this metabolite. So using the CRISPR tool, Cole was able, and this is the guide RNA sequence that he designed, he was able to knock out this specific transcription factor. And later he conducted a transcriptomic analysis to confirm that the rest of the genes are also turned off when this specific transcription factors is deleted. And yes, we actually confirmed that all the genes in the cluster were significantly downregulated in those mutants. We also conducted metabolomic analysis to confirm that this cluster actually belongs and encodes for apenine by running it through a standard that is the apenine commercialized standard. And as we can see here, the wild type is able to produce apenine, whereas the three independent mutants that Cole has selected did not produce apenine on both tested media, PDA and GMM, confirming that this cluster is encoding for apenine and all the genes are downregulated when the specific transcription factors is knocked out. We also later conducted pathogenicity experiments. So similar to what we done in the ECP2 mutants, we conducted those pathogenicity experiments by spraying different popular genotypes with the spores collected from the wild type and the mutants. And to our surprise, we were expected to see a significant downregulation of the disease severity as well as the cankers per centimeters. But to our surprise, we did not see any differences between the wild type and the mutants inoculated plants, which actually kind of contradict our original hypothesis that this compound could be implicated in pathogenicity. But since apenine is already characterized as an antifungal compound, we wanted to look at, since this metabolite is not implicated in pathogenicity, what is actually the ecological function of this compound? Is it really regulating the microbiome? So to test this hypothesis, Cole has selected different fungi that are associated with poplar. Here I'm showing just results from two fungi that were selected and were isolated from poplar. We have Licaria bicolor and Trichoderma gamsii, which are beneficial fungi. And as we can see, when the fungus is growing alone, we were able to measure the growth. And we also did pairwise interactions or co-cultures where Cole grew his mutants and his wild type along with the wild type of Trichoderma gamsii and Licaria bicolor. And as we can see, when the apenine is not produced by the mutants, we did not see any reduction of the growth. Whereas when we grow it with the wild type and apenine is actually produced, we saw a significant reduction of the growth of both fungi. In Trichoderma and Licaria bipolar, we saw significant decrease of the growth. And then here, after that, we confirmed that adenine is not a virulence factor. We were able to use this CRISPR tool and characterize an ancient biosynthetic gene cluster that was found to be providing a competitive ecological advantage to our pathogen over other poplar-associated fungi. So we also, besides looking at anti-smatch, predicting what is absent, what is present in some of those 122 isolates of S. missiva, we used also another approach to identify secondary metabolites that might be implicated in pathogenicity. And this approach relies on modulating the expression of global regulators. So as we have like specific transcription factors, we also have a global regulators or global transcription factors. And they are commonly found in all fungi, and they regulate the production of secondary metabolite by regulating the expression of specific biosynthetic gene clusters. And a lot of those metabolites were found to be implicated in pathogenicity or in the interaction with neighboring microbes. And in the past, when I used this approach in my PhD and in my postdoc, I was able to identify different metabolites that were able to either playing a role in regulating microbiomes or in pathogenicity. So in this first study, when I did a knockout of LA, which is a global regulator, I was able to identify pseudothene, which is a compound that I was able to find that is playing a crucial role in regulating the cheese rind microbiome. And a different study, when I did it on a Penicillium expansum, which is a post-harvest plant pathogen, and I did a knockout of another global regulator, CRE-A, I was able, as we can see on this apple, the mutants were nearly avirulent. And when we did metabolomics and transcriptomic, I was able to identify two biosynthetic gene clusters, one that is encoding for pathogen and the other one for citrinine, that were essential for pathogenicity. So we decided, since this approach has been proven to be very effective in other pathosystems, to use this approach on Spirulina missiva poplar pathosystem to see if that would help us pinpointing to certain metabolites that can be implicated in pathogenicity. So we did a knockout of LA in S. missiva. And when we conducted pathogenicity experiment, we showed, as we can see here, a significant reduction of leaf disease severity across all the genotypes tested, except for one. And we also saw a reduction of cankers per centimeters in certain genotypes, but not in the others, which indicate that that could be an actually very effective strategies to looking at or identifying or characterizing biosynthetic gene clusters implicated in pathogenicity. And we're taking this to doing metabolomics and transcriptomic analysis that would help us seeing what are the clusters that are downregulated in our LA mutants and what are the metabolites that are not produced, which could be like good targets for pathogenicity. And of course, later we have to do a lot of other validations like what I did in my previous studies. So while we are continuously working on identifying genetic determinants of pathogenicity that could serve as targets for the development of the gene drive, we actually decided to start working on advancing this research and actually started developing the gene drive on, I'm going to talk about it later on, the PKS gene of the melanin pathway. But first, what is a gene drive? So when we have a regular Mendelian inheritance and we have an altered gene, this gene would only have a 50% chance to be passed to the progeny. And therefore, after many populations and multiple progenies, this gene would actually be diluted and not present anymore. Whereas when we have this altered gene combined to the Cas9 and the guide RNA, which what we called a drive strain, during the mating, the Cas9 would be able to generate a cut because the guide RNA will guide the Cas9 to this specific region on the wild type chromosome. And then this cut would be repaired by homology-directed repair, transferring this drive machinery to the wild type chromosome. And therefore, the wild type strain would also becoming a drive strain. And that gene would have a nearly 100% chance of transferring to the progeny. So that's how a CRISPR-Cas9 gene drive function. This tool has been widely used in disease vectors to eradicate diseases like malaria and Zika. So it has been developed on mosquitoes and other disease vectors, but it has never been done before on a filamentous fungal pathogen. But to be able to develop a gene drive, the organism of interest has to be having a sexual reproduction or ability to mate. And as I mentioned already in the spirulina life cycle, if you remember, it has actually part of its life cycle when it's on the leaves, it has a sexual reproduction stage. Therefore, this approach can seem particularly promising for effective population control in this organism. So as a proof of principle of the gene drive applicability, before we actually go right away and test it on genetic determinants of pathogenicity, which is our overarching goal, we decided to start working on a gene that would allow us to see an easy phenotyping. Therefore, we decided to target the melanin pathway, because when the PKS gene or the backbone gene of the melanin pathway is knocked out, the fungus would have an albino phenotype. So instead of looking dark green or black, it would have a white phenotype, and that would be easy for us to screen the progenies coming from this mating. So we decided to target this gene. And there's something I did not mention earlier. When we have an active gene drive, when the Cas9, the alter gene, and the guide RNA are localized next to each other on the same locus, we have what we call an active gene drive. So even if it's generating traits that are unwanted, it will be so active and it will spread over multiple generations. So in this case, since it's a proof of principle, we didn't want to design an active gene drive. So we decided to design what was called a safeguarding gene drive. So we decided to insert the guide RNA at one locus, which would be our PKS gene, to interrupt its expression. And we decided to insert our Cas9 at a different locus. And that way, the gene drive won't be active enough, and it will be kind of like safeguarded and contained. So this is how our design for the gene drive are. So first, we're going to do a transformation to insert the guide RNA at the PKS locus. And we have another transformation on the strain to insert the Cas9 at the pure G locus, because it will allow an easy screening on FOA plates. But a crucial problem for synthetic gene drive is the evolution of natural resistance against them. So if we have a mutation in the wild type strain at the guide RNA site, the guide RNA won't be able to hybridize, and therefore the Cas9 won't be able to cut. So as a first step forward towards the establishment of this gene drive, we decided to look, since we have 122 isolates that are sequenced, we decided to leverage from that, and we did an alignment of the PKS gene. And we were able to design 3 guide RNA that are on conserved regions, which can help us predict what's going to happen in nature, making sure that those guide RNA are not in regions that have natural mutations. And we later tested those 3 guide RNA on a substitute of this population, because it's a lot of work to test it on all 122 isolates. So we selected based on this tree, a subset of this population. So we tested them on 29 isolates selected across North America. And when we tested those guide RNA, so how does the test work? We actually sequence a region where the guide RNAs would be able to cut. And then we hybridize the guide RNA and the Cas9 to form a complex. And we use them kind of like a restriction enzyme to see if they will cut in the sequence or not. So it's kind of like an in vitro test for the ability of the guide RNA to cut. And as we can see on this table, guide RNA1 was able to cut in all the tested strains, whereas guide RNA2, although it is designed in a similar way, considering efficiency, specificity, and other criteria that are essential for guide RNA design, it did not cut in any of the strains. Guide RNA3 was able to cut in almost all of them, but not in all of them. So from this quick screening, we were able to see that guide RNA1 is the actual best candidate to go further with in our gene drive design. So Josh was able to take this guide RNA1 confirmed sequence. Oops, sorry for that. Okay, so after confirming that the guide RNA1 was our best candidate, Josh took this guide RNA sequence and he designed a construct that will cut in the PKS. And he performed the first transformation. And here I'm showing some of the plates. He's actually still screening them by PCR and whole genome sequencing, but just wanted to show how the phenotype of the wild type looked like. So it's like, sometimes it's even darker green than that. That's how the wild type looked like, and that's how the PKS phenotype looked like. And as you can see, like, they have a weird phenotype, which is we're really excited about, because that would help us later on in the screening of the progeny once we do the mating experiments. So those strains, after Josh's gonna confirm them that they are correct, they have no Cas9 off targets and anything like that, he will use them to introduce the second components of the gene drive, and the strain would be ready to be tested in our mating assay that we're actually working on developing. So just to give you a brief background on how we come up with this mating assay, we were first able to see if we were able to mate the S. mischieva strains in vitro, but we were not successful. We tested the mating on detached leaves, but also that was really challenging because the leaves would dry out and the mating was unsuccessful. Until we came across this bioarchive manuscript in Zymoseptoria species, which is a closely related species to S. mischieva. And if you remember in the life cycle where we showed that the sexual reproduction actually occur on fallen leaves on the ground where the sexual fruiting bodies would develop. So this assay, actually, we're trying to mimic the natural conditions for mating. So we will use plants. We will spray them with two opposite mating types. And we will let the plants go through their natural senescence. Then we will collect those leaves and we will put them under sterile conditions. We'll use like Velcro to have the plates upside down, plates that have the selection for our genetic events. And then those sexual spores will be able to shoot, and the colonies will be forming on those open plates. And then later we will be able to look for the ascospore-derived colonies under the microscope, and we will perform whole genome sequencing to confirm the mating and the success of the drive. So this mating assay is actually ongoing right now. We don't have results from that yet, but just wanted to show you the approach that we're following, where we are actually really mimicking the natural conditions for sexual reproduction in this pathogen. And with that, I would like to conclude. Hopefully I was able to show you that how we were able to advance the development of CRISPR-Cas9 genetic tool in this non-model organism, how we used this CRISPR tools and how we are still leveraging this tool to pinpoint to crucial genetic factors influencing disease severities, including effector genes and secondary metabolites. How we are taking that a step forward towards the development of the first CRISPR-enabled gene drive aimed at eradicating S. mischieva to protect and sustain poplar plantations. And although this approach offers insight mainly on the S. mischieva populus pathosystem, but it has the potential to be applied to other pathosystems, either in forest ecosystem as well as in agricultural settings. And with that, I would like to thank the CSFA for funding this research. I would like to thank our collaborators at Oregon State University. I would like to thank my collaborators at ORNL and my research group. And I would like to thank you all for your attending the seminar, and I would take any questions you might have.
Thank you so much, Dr. Tannous. That was wonderful.
Thank you.
We're going to open it up to questions now. So please feel free to either raise your hand, directly unmute, raise your hand, or type in the chat and I will read. Go for it, Gillian.
Thank you very much. Can you compare CRISPR-driven homologous recombination with non-CRISPR approaches to homologous recombination?
You mean when we use a CRISPR for homologous recombination, where we don't use CRISPR and we use like a standard transformation?
Sorry.
Is that the question?
Yeah.
Yeah, so in the past, actually, before joining ORNL, I was always using a standard transformation where we design our construct that have homology arms of 1 kb to the region that we want to transform. But after using CRISPR, I really see a great improvements in the specificity, because before, when I was using without the CRISPR to generate the cut, our construct, although designed to be specifically going to the specific region, we saw that it was inserted in other multiple places in the genome. So when we perform either whole genome sequencing or Southern blot, we saw that our construct is like being inserted in different other places. CRISPR has really improved the specificity because it actually generates a cut, and most of those donor DNA that we're inserting in our transformation would go towards this cut to repair the DNA double-strand break. So yeah, we saw a great improvement in specificity. And after we do a whole genome sequencing, we rarely find insertion on other loci.
Thanks. Second question, have you, regarding mating, have you tried mating both partners on senescent poplar?
So take senescent leaves and try it on?
Yeah.
Yeah, no, we did not try that because yeah, that's a good question. But there's another thing that we tried and I did not mention here. We actually think that there's something coming from the leaves, either an exudate or something that is probably secreted during senescence that could be driving the mating. So it was actually Tomas's idea. He took senescent leaves and he did a metabolite extraction by using organic solvent. And he was able later on to lyophilize this crude extract from senescent leaves to see if there was any metabolites that could be secreted and be essential. And he used it in an in vitro setting. So he used those crude extracts coming from senescent leaves into a media and try to do a mating. But we're still not successful. So I don't know if it's actually really the plant material that is essential, not just metabolite that could be on those senescent leaves. But yeah, we did not try it on senescent leaves. We decided to go with the actual natural senescence going on rather than like take senescent leaves and spray them. But that can also be an approach to take.
Thank you.
I have one so it's not directly related to your extraordinary scientific content, but one of the things I was wondering if you might reflect on for the sake of our graduate students in the audience, what your experience is working at a national lab environment. You've had diverse experience at different types of universities, and I was just wondering if maybe like to their perspective, they're exploring different career options. What's it like to work at a national lab, and how might they best prepare themselves for that career option if they're interested in pursuing it?
Sure, yeah. I was actually so interested in academic positions before joining ORNL. I did a postdoc at UW, which was like, yeah, an academic postdoc. So after working at ORNL, I really liked the environment and the dynamic. The nice thing I really liked about ORNL, and I believe a lot of national labs are the same, is that we have like within, so under the same umbrella of ORNL, we have different expertise. So we have, so we're within the bioscience, for example, we have the computational science, we have the analytical group, so they do all our LCMS. We have like the genetic group. So that really helped instead of going and seeking for external collaborators. To be able to do everything under the same umbrella and same institution. And that, like, we find that it saved us a lot of time and a lot of efforts. And it's kind of like each one would focus on its expertise, so you don't have to be doing everything. sometimes for certain people it's good, for certain people they like to be doing everything from A to Z. So in academia, for example, I was the person doing the genetic, the analytical, the pathogenicity experiments, and sometimes like the bioinformatic and computational. I feel like at ORNL specifically, we have this like inter-teams collaborations where everyone works towards the same goal. So within the CSFA, for example, there's a teams of bacterial geneticists, fungal geneticists, fungal biologists, computational science, analytical. And that's like within one science focus area. And a lot of other projects are the same way. And we find that that really advanced the science in a speedy way. I don't know if that answered the question.
Oh, it totally does. Awesome. Is there anything that you might recommend for a graduate student, perhaps, or a postdoc that might be interested in pursuing a career at a national lab like Oak Ridge? Is there anything in particular you'd recommend they do, or reach out resources?
So we all, like for a graduate student, they would have to be associated. So we're all associated with the University of Tennessee, Knoxville, to be able to take a graduate student. So they will take their classes at UTK, but they do their research at ORNL. For graduate students,
I meant like if they wanted to be in a position like yours one day, like what they might do.
Okay, as like a postdoc, for example.
Yeah.
So we always have like postdoc positions open. Sometimes when we go to conferences, and we have a position that is open, we'll try to talk about it and what are the expertise. Then you would apply to every open positions. There's a lot of positions that comes out every week, not necessarily in our division, but there's several directorates at ORNL, and each one would have open positions. And as a postdoc, usually it's for 2 years up to a renewal for a 3rd year. In terms of transitioning from postdoc to staff scientist, it's not always like it happened, because at ORNL, they usually don't hire people that do the same thing. So for example, for me and Tomas, we both are fungal people, but each one of us actually does different things. So to be able to be hired as a staff scientist, you really have to distinguish yourself from other existing scientist or staff scientist. So where Tomas, for example, he does not do genetics at all. I'm the only fungal geneticist at ORNL. I don't know if that answered the question.
No, I certainly think so.
Yeah.
Thank you. And I think we have time for one more question from the chat. We have Isabel Jacquemont Parks asks, she says, "Hi, amazing talk." I agree. "I really appreciate your insight. You mentioned there are some resistant poplar genotypes. Do you know if CRISPR is being used for the poplar genome to address pathogens rather than the fungus?"
Yeah, actually, the research on the plant is way more advanced compared to fungi. And that's what I mentioned earlier. We have a scientist at ORNL that does a lot of CRISPR work on, they're actually developing biosensors right now to be able to detect the pathogens at early stages. So yeah, the research on the CRISPR on the plant part is way more advanced. Whereas when I joined as a postdoc, we did not have any genetic tool to even manipulate. The fungus was really a non-model organisms. Even if you look in terms of publications, there was barely 3 publications on the pathogen. So it was really challenging at the beginning to start from scratch. Whereas for poplar, the research is on another high level.
Wonderful. Thank you so much for joining us.
Thank
you.
you.
For a wonderful presentation. So
thank you so much.
Join me in either round of applause or just kudos.
Yeah.
It's weird.
And please, if anyone have questions or something that maybe I did not touch on during my presentation for the limitation of the time, please feel free to email me, and I would be happy to answer any questions you might have.
Wonderful. Thank you again.
Thank you so much, Katie and Gillian and Lori, and thank you for everyone for attending.
Thank you.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.